Good evening and welcome to tonight's event. I'm Professor Mark Cragg. I'm the Chair of Experimental Cancer Biology here in the Centre for Cancer Immunology. On behalf of the University and the Centre, I'd like to extend a warm set of greetings and welcome to all of our alumni, supporters, staff and students, and those interested in the work we do here in Cancer Immunology. So what I'm going to do is I'll be your host for the evening and I'll be guiding you through the next hour or so. And so give you a brief overview. What we're going to do is I'll give a short introduction into cancer immunology and immunotherapy and really describe what they are. We'll talk a little bit about why we built the centre. And here I'd like just to add a big thank you to all of those that contributed to that. The centre was entirely funded by philanthropy and we wouldn't be here without those fantastic donations. I'll talk a little bit about the success of cancer immunology, but also the challenges and the ongoing issues that we're trying to face in the centre. And really that brings us nicely onto the need for new ideas. And that's where we'll then have two talks by my esteemed colleagues, Dr. Shia Lin Lee and Professor Gareth Thomas, who will tell you both about ideas that they're trying to develop to get for a better understanding of cancer immunology to improve treatments. And Sheah-Lin will give you an overview of a project that's fairly young, and Gareth will give you a bigger breadth project of starting with a small idea to show you where he's taken that and the fantastic developments he's made over the last few years. So at the end of that, I'll give a brief summary of what I think we've tried to tell you over the evening, and then we'll move on to a question and answer session. So the question and answer session, there should be a box to your right and you're able to put in your questions in that box as we go through the evening. So please feel free to ask your questions. We'll be incredibly excited to answer very difficult questions at the end of the evening. So please feel free to put those into the box as we go on. So I'm going to start by really just introducing the main concept. So what we're talking about is cancer immunology. What is that? So that's the study of how cancer and the immune system interact. What it aims to do is identify which immune cells interact with tumour cells and why they do that, to understand how the tumour cells and the immune cells interact, and lastly, to understand how tumours evade the immune system to grow. And that's really one of our largest problems. And this slide hopefully will give you the reason behind that. So the image on the left is really showing you what our current understanding of cancer is, and that is that cancer cells actually don't exist on their own in tumours. They're actually typically surrounded by lots of other normal healthy cells. And actually that's because during our normal everyday life, cancer cells are being detected and removed by the immune system. Unfortunately, occasionally cancers will take on additional mutations, which means they can evade the immune system. And that allows them to grow. And that's when they present into the clinic, when they've actually evaded the immune system typically. And what we try to do then is re-engage the immunosurveillance and the immune system to go back into those tumours and destroy them. And so there's a whole host of different cells involved in that process, a lot of different immune cells, but perhaps one of the most important are T cells, which can go into the tumours and actually destroy them. So destroying them and using the immune system to fight off cancer is what immunotherapy is. And it's using the immune system to treat disease. So it's not specific to cancer, but here in the centre, we're using it to treat cancer. And what we're trying to do here is really help the immune system re-recognize the cancer as being foreign. We're trying to then stimulate those immune cells to respond against the cancer. And a lot of that involves removing inhibition of the immune system that allows the tumour to grow. So unfortunately, what tumours tend to do is they suppress the immune system in a number of sophisticated ways. And what we try to do is circumvent those to allow the immune system to re-engage again so it can attack the tumour effectively once more. And so this idea is not a new one. It's been around for over 100 years. And with many people, they consider William Coley kind of the father of immunotherapy. And that's because he noticed back in 1890s that when some of his patients, their tumours spontaneously regressed, and he noted that there was a correlation with them having various bacterial infections. So those observations, I think, had been made previously, but what he did is he tried to turn that into a treatment. So what he did was actually take the pus-filled bandages from some of these patients, extracted those various pusses and made what he called Coley's toxin and injected that into cancer patients. And he saw some successes, and this was really the birth of cancer treatment in terms of immunotherapy. He also killed a number of his patients because he was injecting quite inflammatory materials into them. But nonetheless, it was the first incidence of demonstrating taking an immune product and generating a response against cancer. And since then, really, we've tried to weaponize the immune system to fight cancer in lots of different ways. Probably these approaches have developed mostly over the last 20 years or so, but you can see surrounding the body here lots of different immune cells, so T cells I mentioned already, NK cells. So these are cells that are a natural part of our immune system. We can take them out of the body, we can expand them and put them back into people to get better immune responses against cancer. We have molecules that are part of the immune system, such as cytokines, which can stimulate the immune system to go and do that again. There are lots of ways we can try to vaccinate the body against cancer. And in the post-COVID era, this is something that's really gained traction as an attractive thing that we might be able to do more successfully in the future. And one of the real successes has been where we've used antibodies, which are one of the effector molecules of the immune system to treat cancer. So they can either directly bind to the tumor or they can stimulate the immune system in the form of immunomodulatory antibodies shown there at the bottom. But the principle here is still the same throughout. We're trying to stimulate the immune system to go back and fight cancer. And that brings us on to a question about why do we need immunotherapy? Why isn't radiotherapy and chemotherapy enough? And this slide is a survival curve for lymphoma. And what it shows you is 4 different types of chemotherapy, which are increasingly more intensive. So they have higher side effects, but unfortunately, you can see that the survival curve doesn't change. So this tells us that just making more and more effective and toxic chemotherapy is not going to improve response rates. So we need something different. And that's what immunotherapy brings us. And that was really the motivation for building the centre and opening it in April 2018. And it's been incredibly rewarding to be part of the field in immunotherapy over the last 20 years or so, to show that immunotherapy is now starting to be successful, i.e. it's starting to treat cancers in a proportion of patients successfully. And so it's hitting the headlines in the general press and in the scientific press, as people can see that it's a way that we can improve treatments for patients. And I'm going to give you 2 very quick examples. One of them is an antibody which targets the tumor cells directly, which is rituximab. So this antibody is used for treating blood cancers. And the graph at the bottom shows you that over the last few decades, the mortality, so the death rate of lymphoma, has actually been rising, even though we got new different types of chemotherapy. And then in 1997, we licensed rituximab. And what you can see since then is actually the mortality has started to go down. And that's even though more people are actually getting lymphoma over that period of time. So that's been a real success. And then the second success has been with these immunomodulatory antibodies. And these antibodies take advantage of the fact that the immune system is really very tightly balanced between positive and negative signals. And that means that we can perhaps move the immune system in the direction that we'd like to do it to get better effective treatments. And so what tumors do is they basically push the red arrow, they suppress the immune system, and they upregulate molecules such as shown here, CTLA4, which is an inhibitory receptor. And so what we then try to do is use antibodies to block that receptor, removing the negative signal and allowing the T cells to then go on and attack the tumor much more effectively. And these particular types of drugs have been very successful, and they've been able to improve responses for certain types of tumors very successfully, such that probably half of our patients with advanced and terminal cancers show some improvement with immunotherapy. We have about 20% of patients who go on to be cancer-free. And if our patients can get past 2 years, then about 90% of those patients can be cancer-free. So that's incredibly encouraging. But obviously, what we want to do is improve that 20% much more than we currently have. And so we need to make immunotherapy even better. And the question is, how do we do that? And really, one of the key findings over the last 10 years has been when we look at our tumours, they can either be hot or cold. And that really means whether there's lots of active immune cells in the tumours or not. And the hot tumours essentially respond to our current immunotherapies quite well, whereas the cold tumours really don't. And what we need to do now is research into those cold tumours and find ways to make them much more susceptible to treatment. And so one of the cells there you can see there is a fibroblast, and that's the subject of Gareth's research. And he'll tell you about how the fibroblast is able to prevent T cells working very effectively. But you can see clearly that there's lots of research to do and find ways to really reinvigorate the immune system, but all of those require a better understanding of cancer immunology, and we need new ideas of how to tackle them. And that leads us hopefully very nicely onto our first talk, which will be from Shia Lin Lee. So Shia Lin is going to give us a talk about the research she's been doing over the last couple of years. So Sheah-Lin is a trainee surgeon and also working in the School of Cancer Sciences as a researcher. And I'm very keen to hear about what she has to tell us over the next 10 to 15 minutes. Thanks very much, Sheah-Lin.
All right, okay. Right, hi everyone, good evening. As Mark has told you, I'm Charlene Lee. I'm a surgical registrar as well as a postdoctoral research fellow in cancer immunology here in University of Southampton. It's really great to see all of you here, and thank you for giving me this opportunity to talk about my experience as well as my research at the moment. I'm sure a lot of you will have heard of the term bench to bedside, which describe how we translate what we find in the lab directly for patient benefit in the clinical setting. But what I'm going to do this evening is actually bring all of you one step back to the step that comes before that, especially for somebody like myself who is a clinician scientist. Because, and I'm going to talk through how I as well as all the other clinician scientists, how we use what we have learned and what we have observed through our interaction and our treatment with the patients, and how we can actually bring those problems that we are facing, the challenges that we face in the clinical setting back to the bench where we can actually, guide what we do in research. So a little bit about myself to start with. While I'm wearing my clinician hat, I'm a general surgical registrar, and through my work as a general surgeon, I do come across a lot of patients who have bowel cancer, which is a research interest of mine. We see this patient when they develop the symptoms. We diagnose these patients. We're often, most of the patient who unfortunately had bowel cancer will have come through and see a surgeon, because a surgery is actually part of a treatment, and it's the mainstay treatment for majority of the bowel cancer. And we do offer quite a good cure rate for bowel cancer. And throughout this interaction with our patients, we are able to identify a lot of clinical problems that I was able to bring to the lab as part of my job. So a little bit about bowel cancer to start with. Bowel cancer is a cancer that develops at the last part of your bowel. It is the bowel that comes after the appendix, and it goes all the way around to the anus. And what has been really interesting for me throughout my career is that as I learned more about bowel cancer, I slowly realized that even though you can have a lot of patients with exactly the same symptoms developing a cancer at exactly the same location in their bowel, when you really dissect all these little cancers down, their tumor microenvironment, which Mark has kind of described earlier, as well as their genomic makeups, are all very different. So really you can say that actually no two cancers are the same. So this becomes a challenge when we treat our patient using the standard therapy of care, which works for a high number of patients, but we do have a significant number of patients who are either resistant to the treatment that we have. They have got cancer that has spread beyond the bowel, which are very difficult to treat. And we also have about 20 or 30% of our patients who initially responded really well to the treatment that we give them, but then through the follow-up system, we then pick up that actually the cancer has come back or the cancer has actually spread further. So these are the challenges that we are facing in the clinic at the moment for bowel cancer. All of you know about immunotherapy. I presume that's why you're here joining us this evening. One of the most effective immunotherapy that we are using at the moment for solid cancers is the checkpoint inhibitors, which harness the T cells' ability to kill cancer cells. And we have heard about how successful it has been in other types of cancer, such as melanoma, such as lung cancer, and kidney cancer or breast cancer. But what about colorectal cancer? So I've made a table here describing which kind of lay out the published study that have used checkpoint inhibitors in colorectal cancer from 2015 onwards. At the initial phases of the study, we are using these checkpoint inhibitors in very advanced cancer that has spread, where we are recruiting patients who has had a lot of treatment failures prior to being treated with immunotherapy. And you can see even then at the top part of the rightmost column that the response rate is actually about 40%, which is really not bad for this group of patients. They are very challenging to treat. As we become more experienced with using checkpoint inhibitors, and as we experience more successes using them, we start giving checkpoint inhibitors to patients with colorectal cancer who has an earlier stage of disease. And actually what we are seeing is that even at a very early disease stage, the response for checkpoint inhibitors are really good. And one of the studies that I wanted to point out from this table is actually the last study that was only published last year in 2022 persisting in New England Journal of Medicine. This is a study that is conducted on rectal cancer, which for us remains very challenging to treat because a lot of these patients will have to go through chemotherapy, they have to go through radiotherapy, and then they can have a life-altering surgery, in the sense that even if we have successfully cured the cancer, a lot of them will never have a normal bowel function, and a significant number of patients who has rectal cancer at the very last bit of their bowel could end up with a permanent colostomy, which, although it's not life-threatening, is definitely very life-altering for the people who have to go through this. And what this study has shown is that if you give this group of patients with immunotherapy, even though the study itself has some big limitations in the sense that there's only 12 patients in the study, and the follow-up period is very short, we are seeing very, very positive response, which is really encouraging for those that are working in cancer immunology in colorectal cancer. However, this really effective treatment has a caveat in colorectal cancer, in the sense that only a very small subset of patients at the moment in colorectal cancer can benefit from this therapy. And sorry, let me go back. And the subset of colorectal cancer who are actually benefiting from checkpoint inhibitors at the moment are the one that has the phenotype called microsatellite instability. And only about 14% of colorectal patients have this phenotype, whereas the vast majority of patients who has another phenotype called the microsatellite stable cancer are not able to reap the benefit of these very effective drugs at the moment. So I think, you know, from a clinician point of view, not being able to offer a very good treatment for majority of the patient that you see is quite challenging for myself. And I think that's why I really appreciate the scientist part of my job, because when I have a problem that I see in the clinic, I can then bring it back to the lab and see what can I do about them, and whether I can generate any new ideas, whether I can test them. And throughout this process, I'm able to learn a lot of new skills, a lot of new methodologies, and also form different collaboration with different groups in the university to and see how we can use all the expertise available to us to solve the problem that we see. So coming back to the problems at hand. So one of the first thing I thought about when I go to the bench is whether these two phenotypes are truly distinct phenotypes, or whether actually there's an overlap within this phenotype that we can exploit, in the sense that if there's any features that are common between these two, can we select those patients with a microsatellite stable phenotype and trial them with immunotherapy and see whether they will respond just as well as the other group. So with the funding that I've received from Cancer Immunology Fund, as well as the already established infrastructure that I have in University of Southampton, once I've generated this idea, I was actually able to very quickly translate this idea into experiments. So what I've done is I have selected 30 colorectal cancers where from the tissue bank that have microsatellite instability phenotype. And I was able to do a couple of different analyses on them, looking at not just the quantity of the T cells, but also the quality of the T cells response, which is really important when we're talking about response to checkpoint inhibitors. I was also able to actually have a good overview about the tumor microenvironment in this group of cancers using the fund that has been provided very generously by our funders. And Mark already alluded to the importance of studying T cells. And what I just wanted to point out is that the T cell is actually a very unique immune cells in the sense that those two little antennas that you see on your screen, they are actually called the T cell receptor. And the way it functions is almost like a lock and key function, where the T cell will only, majority of the T cells, I should say, will only be activated if it's able to see an antigen that fits into the groove on the T cell receptor. And the antigen can come either from virus, it can come from cancers, but that makes it a very unique kind of activation method, so that we don't get just a rampant immune response. And looking at the 30 colorectal cancer samples that I have with microsatellite instability, I was actually able to make a very interesting observation in how variable the quality of the T cell response is in this subgroup of cancers. So on the left-hand side of your screen, you'll be able to see a cancer will actually have a good T cells response, because you can see that it has got a lot of T cells, but there are multiple copies of the same T cell, which means that this is a cancer where the T cells inside the microenvironment is able to see an antigen, is able to, it has the right environment for the T cells to actually be activated and proliferate and able to exercise its cell killing power. Whereas on the right-hand side, you see a cancer that even although it has got the same number of T cells, is actually made up of multiple different copies of T cells. So what we think is that this cancer is not able to mount a very good T cells response. So you have got a lot of T cells sitting in the cancer, but they are not being, their potential and their real benefit are not being exploited. And this is really important because from our experience with melanoma, which is has been very successfully treated with checkpoint inhibitors, we know that the tumor that has a good T cell response has a much higher chance of being responsive to checkpoint inhibitors, and hence leading to a more durable response and cure. With my collaboration with one of the senior bioinformaticians in our group, Dr. Stephen Thirlborough, we are also able to actually find out a unique biological pathway that is closely associated with a good T cells response. And what has been really encouraging actually is that we are able to see the activation of this biological pathway in the subset of colorectal cancer with microsatellite stable phenotype. So I think the next step for us is actually to try and dissect this biological pathway out and to find a way to first to identify this subset of cancers, which might be able to accelerate into a clinical trial where we can trial them with checkpoint inhibitors and see whether they will have the same response as colorectal cancer with microsatellite instability. So this is my research at the moment is at the very early stage, but I'm hope and my aim is that for the next couple of years, what I will be able to discover will lead to better diagnostic and prognostic processes. Well, I would like it to be able to stratify patients even better from what we have at the moment in the clinic, so that we can give patients the treatment that they are most likely to respond to in the quickest way possible. And also, throughout the research, I'm sure Gareth will talk a lot about this, is that we'll find out a lot of different new things about the tumor microenvironment in colorectal cancer, which will hopefully open the doors out for a lot of new therapies as well. That's all I've got for this evening. Thank you everybody for coming, and enjoy the next talk.
Fantastic, thanks Sheah-Lin. That was a great overview, and I hope people can see that actually from a relatively small investment in that research funding, Sheah-Lin has already found something that we didn't know before about T cells and actually what's going on in some of those cancers, which is something that we can now, hopefully she can look forward to developing in the coming years. And so now we're going to build onto a more mature story. So Professor Gareth Thomas is our next speaker. So Gareth is a clinician, is a pathologist, but also a tumor biologist, and his main area of expertise, amongst many others, is fibroblasts, which are a particular cell type which are often seen in some of these tumors. And I think Gareth's going to give you an overview of what he's been doing over the last number of years, which I think will give you a really nice perspective of kind of taking an idea and developing it fully through to some significant findings. Thanks very much, Gareth.
Thanks, Mark. More mature, Mark, just means I'm older and over the hill. Good evening, everybody. It's a real pleasure to be speaking with you this evening. I'm Gareth Thomas. I'm Professor of Experimental Pathology in Southampton. And I'm going to talk this evening about one of the questions that Mark's already alluded to in his introduction. Why doesn't immunotherapy work in some patients? And I'm going to talk about a project where we've taken a basic idea in the lab and we've taken it right through to a clinical trial, which opened this year. It's important to remember that cancers are collections of cells. So it's not just the tumor cell itself, but there are lots of other different cell types within the tumor. So there are blood vessel cells, different types of immune cells, and cancer-associated fibroblasts, which is the cell that my lab is particularly interested in. Some of these cells will, such as cancer-associated fibroblast, which is a sort of a structural cell, which is found in tumors. These can make the tumors behave aggressively, and they're associated with poor survival if a patient has lots of these cells in the tumor. Other cells like tumor-infiltrating lymphocytes, and we've heard a little bit about white blood cells already. If a tumor has lots of these cells within them, then these patients are associated with good survival. And you can see the Kaplan-Meier survival curves. These are just a sort of a statistical analysis of the way that we analyze patient survival. As a pathologist, we can stain for cancer-associated fibroblasts or tumor-infiltrating lymphocytes in patients' tumors. So for cancer-associated fibroblasts, we stain for smooth muscle actin or SMA. And then we can stratify patients according to whether there are high levels or low levels of these cells within the tumor. And then we can generate these Kaplan-Meier survival curves. And essentially, the steeper the curve, the worse survival. And you can see that patients that have moderate or high levels of CAFs in their tumors, actually their survival is very poor compared with those patients that have very few. And it's the opposite way around with tumor-infiltrating lymphocytes. If you've got high levels of T cells in your tumor, in this instance, we've stained for a T cell marker, CD8, then if you've got lots of these cells, then survival is very good. And actually, there's an inverse correlation between these two cell types, which I'll talk about a little bit later. If there are lots of cancer-associated fibroblasts in the tumors, you usually find very few T cells. And immunotherapy, and particularly anti-PD-1 checkpoint immunotherapy, is probably one of the most exciting breakthroughs in cancer therapy for probably since the 1970s, when chemotherapy became used widely. But despite many exciting successes, you can see from the table on the left that there are some tumours where really we get very poor responses to these. And Sheah-Lin has already talked about colorectal cancer. My interest is in head and neck cancer, and we can see that across the board, perhaps only about 15% of patients are responding to this drug. And that begs the question, why doesn't immunotherapy work in some patients? And if we can study this and we can understand the resistant mechanism, can we actually target these to improve response rates? And one of the ways we went about this was we thought we would look at tumours that had very few T cells within them. And we did this using a complex computational approach where we looked at the DNA methylation. This allowed us to deconvolute tumours and to work out what types of cells were present. And we were looking for those tumours that had very few T cells. And then we could look at gene analysis, and we could look at DNA mutation analysis, and we could look for features of immune cold tumours that had very few T cells. And there were some common features, things like KRAS mutations and PTEN deletions. But one of the features which fell out of this was the abundance of cancer-associated fibroblasts correlated inversely with the presence of these T cells. So lots of CAFs in the tumour, very few T cells. So this suggested to us that CAFs perhaps were one way that a tumour evade the immune system, and that if we could study and understand how the CAFs were doing this, then maybe we could target CAFs as part of immunotherapy to make it work more effectively. So what is a cancer-associated fibroblast? Well, a fibroblast is a very common cell found throughout the body. It's a sort of structural cell. You find it in your skin, for example. It produces collagens and those sort of extracellular matrix proteins that are in your dermis. During a healing wound, they become activated. They turn into these myofibroblasts, which are contractile cells. They express markers like smooth muscle actin, SMA, so we can identify them in tissues as a pathologist because we can stain for this particular protein. And their role in a healing wound is they contract the wound and then they produce the scar tissue. So they pump out a lot of collagen and other proteins that create this collagenous scar. And of course, in a healing wound, they're very tightly regulated. So as soon as the wound heals, the myofibroblasts disappear. But they can persist in certain conditions such as organ fibrosis or liver fibrosis or kidney fibrosis. But also you find subgroups of most solid cancers have them. So for example, in head and neck, probably about 50% of patients have cancers with high or moderate levels of CAFs. It's higher in colorectal cancer and esophageal cancer, and in pancreatic cancer, which is sort of the archetypal CAF-rich tumor, which is notoriously difficult to treat. When we grow fibroblasts in the lab, we can turn them into myofibroblasts by treating them with a cytokine called TGF-beta. So we put the fibroblasts in plates, we treat them with TGF-beta, and they become these contractile collagen-secreting cells that we can see in culture by staining them for smooth muscle actin, which is shown by the green stain in the panel on the left. And they also start pumping out more collagen, which is shown in the panel in the right. So these are quite easy cells for us to model in the lab. So we wanted, because we discovered this inverse correlation between fibroblasts and T cells, we wanted to study what the CAFs were doing immunologically. And the problem was that when we started looking at mouse tumours, and classically the ones that we use to test different immunotherapies in, they contain very few cancer-associated fibroblasts. You can see that we've used a stain here for smooth muscle actin, or CAF marker. The mouse tumour has hardly any at all. And if you compare that to an equivalent human lung cancer that has high levels of CAFs, you can just see the difference in how these tumours look down the microscope. So firstly, we needed to generate a new cancer model. So we thought, well, if we want fibroblasts to be present, we're going to have to co-inject these cells with tumour cells to try and develop this same sort of feature that we're seeing in human tumours. So we would take fibroblasts in the lab, we would treat them with this cytokine called TGF-beta for 6 days, and then we would co-inject them with tumour cells into the flank of the mouse. And then we could study the way that these were influencing tumour behaviour. We could measure the tumours and look at whether they were spreading to distant parts of the mouse, different organs, whether they were metastasizing. We could look at the immune response and to see whether that had changed. And we could also see whether the CAFs were affecting response to therapy. So this is an example of that lung cancer model. So in this experiment, on the left, we're looking at growth curves, and we've got a control group of tumours where we've just got tumour cells growing, and then we've got a second group where we've got tumour cells with the CAFs present. And you can see that the tumour cells with CAFs, they grow much larger, much quicker. we can stain for smooth muscle actin, and we can find this very SMA positive calf-rich stroma, which looks a lot like what we see in human patients. And then we started looking at different immune cells. And our most important observation was that these calves were stopping from T cells from getting into the tumor. So when the calves were present, the T cells were getting stuck around the edge of the tumor, and they couldn't actually infiltrate into the tumor and kill the tumor cells. Now, if you consider that most of the immunotherapies which are being developed are predicated on T cell killing of tumor cells, if the T cells can't get into the tumor in the first place, this is a very important barrier that needs to be overcome for most immunotherapies to work effectively. And because of this observation, we hypothesized, as you would expect, that these calf-rich tumors would become resistant to different types of immunotherapy. So TC1 is an interesting model. It's a cancer vaccine model. It's a mouse lung cancer, but it expresses human papilloma antigens, E6 and E7. So human papilloma virus is a virus that causes cervical cancer and also certain types of head and neck cancer. And we can take a vaccine against this antigen. The tumors will grow in the mice, then we can vaccinate the mice. They will generate an immune response against this antigen, and the tumors will clear. So the mouse will mount an immune response against the tumor. If you look at the growth curves on the left, if you just look at the red line, that's the tumor cells growing by themselves, treated with a vaccine. And then if you look at the blue line, the tumor cells with the calves treated with a vaccine, and you can see that when calves are present, we're starting to lose this therapeutic effect that we see with a vaccination. We also wanted to look in a model of which is really the gold standard immunotherapy at the moment, anti-PD-1 checkpoint inhibitor. So for this, we used a different cancer model. This is called MC38. It's a colorectal mouse cancer model. But again, we're just injecting it into the skin of the mouse. And here we're treating, again, there are 4 different groups of mice. And if you just look at the calf-containing groups, which are shown by the blue and the orange line, the blue line is the tumor cells growing by themselves, These are very sensitive to anti-PD-1. So when we treat the mice with anti-PD-1, the tumors essentially clear. But when the calves are present, which is shown by the orange line, then we lose this response. So the calves are stopping this immunotherapy from working effectively. And there have been a number of high-profile studies which have come out in recent years. They've taken patient material, patients who've been involved in clinical trials where they've been treated with anti-PD-1 or anti-PD-L1, and they've looked for genes that are associated with non-response in these patients. And one of the consistent findings is that tumors with high levels of cancer-associated fibroblasts just respond very poorly to anti-PD-1 checkpoint blockade. So this makes targeting calves a very interesting strategy, because lots of patients have calf-rich tumors. Calves are preventing immunotherapy from working effectively. How can we go about trying to knock off the calf population? And you've got different options. You could just try and kill off your calves in the tumor. You could try and understand how the calves are actually functioning to stop the T cells from getting into the tumor and try and specifically target that mechanism. You could perhaps try and stop them forming in the first place. That is, of course, difficult when you've already got an established tumor. But what we've been trying to do is normalize a calf, so to turn a cancer-associated fibroblast back into a normal fibroblast. And that's quite an attractive idea, because we have some evidence that these normal fibroblasts are actually tumor suppressive. So perhaps we could turn a bad cancer-associated fibroblast into a good fibroblast, which may actually help support an immune response within the tumor rather than stopping it. And so we studied what was going on when we turned a fibroblast into one of these activated calves. So we started off with a quite simple experiment. We took fibroblasts, we treated them with TGF-beta, and we did some RNA sequencing to look at gene expression, global gene expression, as to what was going on in these cells as they differentiated into a calf. And to cut a long story short, we identified an enzyme called Nox4, which was strongly upregulated as the cells differentiated. And the role of this enzyme is to produce reactive oxygen species within the cell, which is important during this calf differentiation process. Because this was quite a simple experiment that we performed in the lab, we then wanted to go and look in human tumors to see if we could find a relationship between fibroblast gene expression and this gene Nox4. And we could find that by doing this gene correlation network analysis, which is shown on the right, where we found that Nox4 was very tightly associated with smooth muscle actin, which is our marker of CAFs. And we could see that Nox4 gene signature really sat very tightly and quite specifically within a CAF gene signature. So it looked in human disease as if Nox4 was very tightly associated with the formation of CAFs. And so this was our idea, that maybe we could target Nox4, and if we could do that, then maybe we could get immunotherapy to work better in those patients that have tumours with lots of CAFs within them. So we needed to find a company that really made a Nox4 inhibitor. And at that time, and this was about 10 years ago, we found a company called Genkyotex, who had developed a Nox4 inhibitor, and it was by far and away the most advanced Nox4 inhibitor in the market. It had already been tested clinically. And interestingly, this was only a small company, but they had developed a Nox4 inhibitor to treat fibrotic disease. So they had been testing inhibitor in patients with lung fibrosis and kidney fibrosis and liver fibrosis with some really quite promising results. And it was also a very safe and well-tolerated drug that could be taken orally. What they hadn't considered was perhaps that this drug could have use to treat cancer. And as I've already mentioned, those myofibroblasts that we find in healing wounds and in fibrotic organ conditions, those are very, very similar to the cancer-associated fibroblasts that we find in patients' tumours. And I'm just showing you an example experiment here. We've taken CAFs, we've grown them from a patient's tumour, we've grown them in the lab. We've stained them to show that they're expressing smooth muscle actin, which is what the green staining is on the left. But when we put the drug setanaxib, the Nox4 inhibitor, on them, they downregulate all of these genes which are associated with this CAF differentiation, things like smooth muscle actin, things like collagen, and they turn back essentially into a normal fibroblast. So this drug was effective not only at preventing the differentiation of a CAF forming in the first place, even when a CAF had formed, we could turn it back into a normal fibroblast, and we could reverse the differentiation process. So, of course, the obvious question was then, would this drug have any use in treating cancer? So the first experiment we did, we were just using this drug as a monotherapy by itself. So in this mouse experiment, we've got two groups of mice. They both have CAF-rich tumours. One is just receiving the vehicle control with no drug, and the second is receiving this setanaxib, which is also called GKT. And you can see just by using the setanaxib, the tumours grow much smaller. If you look at the smooth muscle actin staining, where we're trying to look at fibroblasts, you can see that the drug has got rid of this fibroblast-rich stroma. And more importantly, perhaps, is that this loss of this fibroblast stroma is associated with a real influx of CD8 T cells in the tumour. So we've managed to get rid of the CAFs, and that's pulled the T cells back into the tumour. Because we're always a bit worried when we test drugs that we may be having off-target effects, that the drug may be working through another mechanism and not actually working on Nox4. So we can also take an approach where we take the fibroblasts and we knock down the Nox4 gene within those fibroblasts. So this is specific to the fibroblast. And essentially, we could produce the same effect. When we knocked Nox4 down in the fibroblast, the tumours were much smaller. We lost that CAF-rich stroma, and we found much higher levels of CD8 T cells within the tumour. And then finally, we started to combine the setanaxib with with different types of immunotherapy. In the first experiment shown in the top panel, we've gone back to our MC38 model where we test anti-PD-1. We've got 4 groups of mice. All of them are bearing CAF-rich tumours, and then we've tested anti-PD-1 as a monotherapy, the setanaxib as a monotherapy, and then we've put them together as combination immunotherapy, hoping that they'd have a synergistic effect. And you could see that that's actually the case. If you look at the pale blue line and bottom of the growth curve, the mice receiving the combination of much smaller tumours, and they have much more significant survival. And we did the same thing also in our vaccination model, our TC1 model, 4 groups of mice, all containing CAF-rich tumours, some treated just with a vaccine, some just with setanaxib, and some with a combination. And the same thing again, the combination produced much smaller tumours and significantly improved survival, and in fact cured the mice when we extended the vaccine protocol. And this really generated the data to run the clinical trial, which we're very excited about. So this is a trial is now running in about 30 centres across 7 countries in Europe and the US. And this really is looking at patients with recurrent or metastatic head and neck cancer, which is what my clinical interest is. And it's looking at whether we can use anti-PD-1, pembrolizumab, with and without cetuximab. And this is interesting because we're selecting the patients that we think will benefit. Sheah-Lin's already alluded to the importance of having biomarkers so that you know which patients to choose for your particular treatment. So for this particular trial, we're picking patients that have tumours with high or moderate levels of capsin, and we can do this quite simply with our pathology stain for smooth muscle actin. And then these patients will receive pembrolizumab, and some patients will be randomized to receive cetuximab or not. The primary endpoint for this particular trial is we're looking for a change in tumour size. But I think what's important for clinical trials these days is that we're paying much more attention to the analysis of patients' tissues. So each patient is going to have their full transcriptome analysis. We're going to look at tumours before treatment, and we're going to look at tumours after treatment. And we're going to look at gene changes across the course of treatment. And in doing so, we hope we'll identify why some patients may respond and perhaps why some don't. And we're also extending that analysis in a proportion of patients, even down to a single cell level. And we're hoping this is going to give us a lot of biological information that we can then take back to the bench to shape our research there. So just to summarize, so CAFs are tumour promoting. I've spoken this evening about their role in shielding tumours from immune attack, but they actually do lots of other things like promoting metastasis, for example, as well. So they're a very attractive tumour target. And in fact, there's lots of interest from pharma at the moment of trying to develop different therapies that target CAF. They do promote immune evasion. Importantly, they shield tumours from immune attack by preventing T cells from getting into the tumour. And by doing this, they confer resistance to immunotherapy. Trying to normalize the CAF is an attractive proposition. I've already mentioned the potential tumor suppressive effect of normal fibroblasts, but we've shown that at least with our Nox4 inhibitor, we can overcome this T cell exclusion and we can get a much better immunotherapy response in tumors that have high levels of these CAFs. And very exciting. I mean, it's taken us about 10 years to get this point. The clinical trial started earlier this year and is recruiting well, and we're hoping to get some data through on this in a few months. So thank you for your attention, and I'd be happy to take any questions.
Fantastic. Thank you, Gareth. So I hope everyone could see that was a real fantastic example of making an observation that came from the clinic, going into the lab, trying to understand the mechanism, and then coming back to the clinic with a way of modulating immunotherapy. So I think that's a fantastic story that came from an initial idea that the fibroblasts and the T cells maybe were interacting together. So thanks very much for that, Gareth. So I'm just going to give a short summary around immunotherapy. So hopefully we've convinced you that it's an exciting new treatment modality for cancer. It's clear that it's treating an increasing number of diseases, and that's alone and in combination, as you've heard from Sheah-Lin, some of the combinations that have been presented. I think it's key that we understand which patients to treat with which immunotherapy, and that basically comes down to how are the tumours evading the immune system in the first place. And that's all part of personalised medicine, understanding the tumour to treat it appropriately. And it's really only by understanding how the immune system works and sees the cancer can we really harness the immune system better to eradicate the disease for all of our patients. So what's left to do? Obviously, we need to develop new and better immunotherapies. We need to move the dial from treating sometimes low proportions of patients successfully to treating far greater proportions. We need to understand as part of that why immunotherapies fail in some people, and develop new treatments to overcome those different types of resistance mechanisms we see. And obviously, we're always trying to provide better, more specific, less toxic treatments for our patients. And it's clear that, that needs a lot of research and a lot of funding. As Gareth just mentioned, the study that he's talked about is a 10-year endeavour with lots of people involved and lots of experiments. And that really that's where the Cancer Immunology Fund has been fantastic for us over the last 4.5 years or so. It's been very helpful for us to be able to fund people to do some of the research. We've been very fortunate in being able to buy state-of-the-art equipment. If any of you have done a tour with me, I'll have no doubt shown you the black box down there, which is not very big, but costs nearly £200,000. That is very unimpressive to look at, but enables us to look at receptors on the surface of cells in individual detail, individual receptors, and see how they move when we give them antibodies or treat them in different ways. So it allows us to understand the immune system and our cells much better. And last but not least, you know, having these kind of pump priming awards, which Sheah-Lin received one of, you know, it allows us to provide new ideas, to provide new understanding about what's going on in cancer, and how we can perhaps use that knowledge to provide new treatments. So thank you to all of you that have contributed to the Cancer Immunology Fund, and we'll move now to the last section of our set of talks tonight, and that's the Q&A session. It's the bit that we look forward to the most, because we don't know what's coming, so we'll just have a number of questions that come through. If you haven't already, then feel free to just pop your questions in the boxes on the right. I can see that there are a few already in. So I think the first one, maybe Sheah-Lin, if you want to take this one, it is, how does immunotherapy work alongside treatments like chemotherapy and radiotherapy?
Okay, I think first and foremost, why in the clinic, the rationale of us combining chemotherapy or radiotherapy alongside immunotherapy has to do with what we've alluded to earlier today already, is that, you know, although immunotherapy has worked really well for a lot of patients, there is still a significant number of patients who had primary, where they are resistant to immunotherapy from the beginning, or secondary resistance, where they initially respond really well, but the cancer eventually come back, and the immunotherapy actually stop working. And by using a combination of different therapies, we potentially can ameliorate the mechanism of resistance. So, for example, in colorectal cancer, we recently had a trial that has published a result where they have tested a combination of the standard chemotherapy as well as radio, as well as another monoclonal antibody called bevacizumab, together with immunotherapy, and compared them with the group who received the two other drugs but not immunotherapy. And it has shown that by adding on immunotherapy, the patient actually have a higher response rate to the treatment. And also the effect that they are seeing is much more durable. We don't know the exact mechanism of why combining is better, but the theory behind it is that when we treat these cancers with chemotherapy or radiotherapy, it's almost like a mass effect where all the cells that are proliferating would die, and that cell death actually release a lot of antigen, which is what the T cells can pick up and see. And once that first signal has been achieved, if you are in the right environment for the T cell response to be activated and to proliferate, it then makes the immunotherapy much better. And that's one of the hypothesis of how the combination therapy actually works. And it's actually, we're already using this combination therapy, not just in colorectal cancer, we're using them in melanoma, we're using them in renal cancer, as well as lung cancer in the clinic.
Fantastic, so thanks Sheah-Lin. So the next question is definitely for you, Gareth. It's, what's to stop normalized CAFs fibroblasts from being reactivated in the tumor to CAFs again?
That's a very good question. I think as long as you're still using the inhibitor, that would prevent that. So the setanaxib not only reverses the CAF differentiation, but it prevents it from happening in the first place. So I think that would be key. Of course, see, what you're hoping for is that by combining setanaxib with the immunotherapy, that the tumor will then actually clear, so there won't be a tumor there trying to generate, these corrupted fibroblasts.
Great, thank you. So the next question is, what proportion of tumours are cold tumours? And I think Sheah-Lin kind of mentioned it, and I think Gareth mentioned it also, that it really depends on the tumour type. Melanoma tends to be one that there are more hot tumours, so they respond well to the antibody immunotherapies that we have. Some of the others, much less so, and certain types of pancreatic and other tumours, actually, there are much less hot tumours. And what we're finding is that, as Gareth described with the fibroblasts, that's one of the reasons why some of these tumours might have T cells excluded from them, and others just don't have them coming in at all. So the fibroblasts aren't there. And it's really understanding within a tumour type, whether that be pancreatic, prostate, or whatever, actually what the individual tumour looks like, to understand how best we might go about treating it. So it really does depend on the cancer type.
Yeah, and I think, as you said, Mark, different reasons for different tumours. Often tumours with lots of mutations within them, which high mutational burden, they respond quite well to checkpoint inhibitors. Things like melanoma, things like lung cancer, high mutational burden, lots of new antigens for the immune system to recognise. The tumours have far fewer mutations and they tend to respond more poorly. But there are also other reasons, like the presence of CAFs, and pancreatic cancer is a very good example of that. Probably more than 95% of pancreatic cancers are really CAF rich, and they don't really respond to very many types of therapy at all. Very difficult to treat.
Yeah, thank you. And the next question is, I would like to know the success rates for the 10 most common cancers and how these rates are likely to change in the next 5 years. So maybe, Sheah-Lin, do you want to have a go at that one first, as a practicing clinician and a surgeon?
I think, with solid cancers, which is really what I deal with in the clinical setting, the 10 most common cancers are things like breast cancer, prostate cancer, melanoma, as well as lung cancer. So for breast cancer, prostate cancer, and melanoma, the survival is slightly better when you just talk about them as a whole group. Obviously, the survival is going to change with how advanced the cancer is when the patient is diagnosed. So for the better prognostic cancers like breast, prostate, and melanoma, it's about 75% of the patient will be alive in 10 years' time. For bowel cancer, non-Hodgkin lymphoma, or kidney cancer, the survival as a whole group is unfortunately only about 50%. And for the poorer prognostic cancers, such as lung cancer, pancreatic cancer, or brain tumors, the survivor in about 10 years, I think is probably less than 10% off the top of my head. So it really varies between different tumor type, and then within each tumor type, it also varies between the stage of the disease.
Yeah, fantastic. Yeah, Gareth, did you want to pick up on that?
Yeah, on a positive note, I think it's important to remember, that survival rates for cancer across the board, have doubled in the last 40 years. And as Sheah-Lin's already said, there are certain types of cancer which are notoriously difficult to treat with poor survival: pancreatic, lung, esophageal, brain. And these have been identified as cancers of unmet need by Cancer Research UK, and they've doubled the funding into those cancers since 2014, but very variable across cancer types.
Fantastic, thank you. And we've probably got time just to squeeze in one last question. So what are your thoughts regarding the potential for cytotoxics activated by exploiting differences between the tumour microenvironment versus healthy cells? I think that's a great question. That's definitely something that people are trying to do. So tumours are different than healthy tissue. They have all sorts of enzymes and substrates, things that aren't there in normal tissue. So certainly one of the things that is of interest in the antibody field is to use antibodies that only become activated in that particular microenvironment. So if a protease, which is something that can chop up a protein, can cleave a part of an antibody to only activate it at the tumor site, that is something that can be very useful for targeting. And one of the other questions I noticed was asking around toxicity. And sometimes antibodies and immunotherapies can cause side effects, which are immune side effects often. And that's actually one way, exploiting the tumor microenvironment is actually one way to try to reduce those toxicities. But I think it's a great question. So I think we are almost out of time. So we'll probably draw the Q&A to an end there. So I'd just like to close by saying thank you, everyone, for joining us tonight, and for everyone that's generously supported the Cancer Immunology Fund over the last 4.5 years. Hopefully you can see some of the research that we're doing in the centre, and hopefully you agree that it's interesting and important. If you've enjoyed tonight's talk, then I'm delighted to announce there's details of an in-person talk happening on Tuesday, the 23rd of March. That's actually happening at the Wellcome Collection in London. And what it is, it's concerning how AI and data scientists and engineers can actually develop personalised treatments for diseases like dementia, but also cancer. So if you're interested in that, then there'll be details sent through the email channels if people are interested in signing up. And also, there'll be a short poll pop up on your screen in the next couple of minutes, which essentially says whether you enjoyed this evening. If there were bits of it you didn't like, my hosting, I can understand, whatever it is you'd like to feedback. It's really helpful for us to kind of improve these type of events. And finally, if there's anyone who'd like to see this all again, it has been recorded and it will be provided to you over the coming days in your inbox. So thank you very much for hosting. Thanks to my co-panelists for giving up their evenings to present this evening, and thank you all and good night.